And welcome to this Chemistry World webinar. I'm Ben Valsler, Chemistry World's digital editor. My role today is to take you through the software that we're using so that you know how best to interact with our speakers and our topic for today's presentation. So the first thing I'll do is tell you about GoToWebinar, which is the software that you've logged into in order to interact today. And the most important thing you need to know is how to ask questions. Down at the bottom of the GoToWebinar panel, for most people, there will be a box that you can use to send a question to staff. So feel free to ask any questions you have at any point throughout the presentation. What we will do is we'll keep hold of all of those until the end, and then we'll do our best to get through as many as we possibly can. So do please keep your questions coming in. It's really important for us to know what it is that you want to know to make sure that we can answer them for you. It really helps us not only to shape this webinar to be exactly the experience that you want, but also to plan future webinars and make sure that we know what our audience want and we get you the things that we need. Now, speaking of getting things to you, all of the live attendees for today's webinar will receive a certificate. That's our way of saying thank you and proving that you've put your time to good use. I appreciate for most people in the UK, at least, it is lunchtime right now, and you're using your lunchtime particularly well to learn about good practices in data analytics. So do keep your questions coming in. There will also be a couple of polls that we're going to run throughout the webinar. Again, very useful for us to get to know you a bit better. So do please engage with those when they turn up. So the topic and the contents for today's webinar. As I said, we're looking at good practices in data analytics, and we're working with our partners, Jump. We've worked with Jump a great deal. They're part of SAS, which is the leader in business analytics software and services. They work very hard to help scientists, researchers, people in the industry use statistical methods that can make the most of their work, their processes, their practices. And not only do they work very hard to educate people about what the most appropriate statistical methods are, but they produce software that makes using those statistical methods an absolute doddle. And today we're going to look at how that software has been used to particularly good effect with our guest from the University of Limerick. So we'll be learning how the University of Limerick develops skills and knowledge of data analytics through their Pharmaceutical Manufacturing Technology Centre, or PMTC. That will probably come up quite a few times. So PMTC, Pharmaceutical Manufacturing Technology Centre. In a recent collaboration with their industry partners, the PMTC developed a good practice guide to data analytics, which was a conclusion of over 400 hours of interaction with industry across focus groups, case study presentations, and interviews. And we're going to benefit from their experience today. A number of good practices in data management, data visualization, and model development are going to be covered over the course of the next hour. So we're going to learn how data analytics can be defined, why data readiness is very important for you, how data visualization can be used very effectively at all levels in a business or a research group, how a number of good practices which should be considered in model development, and we'll look at opportunities to collaborate with PMTC and access their good practice guide. So once again, thank you to them and thank you for Jump for putting us in touch. We have two speakers today, so we'll see software demonstration and we'll see presentation. Our first speaker will be Marcus O'Mahony, who's a senior research fellow with the PMTC at the University of Limerick. He supports the focus of data analytics within PMTC, is passionate about developing new technologies for pharmaceutical manufacturing. Once we've heard from Marcus, we're then going to hear from Volker Kraft, who is JMP Academic Ambassador for Europe. He's a senior academic ambassador. He's got a background in electrical engineering. He's long used statistical methods to study psychoacoustics and speech communication, and applies his analytics to customer advocacy and telecommunications, previously for Vodafone, Voxio, all sorts of other people. So that immediately, pharmaceuticals and psychoacoustics, give you an idea of the breadth of different areas of research and areas of industry and work that this sort of statistical analysis can benefit. So regardless of what area you personally work in, there's almost certainly some way that you can use these approaches to benefit you in your working life. And we'll hopefully cover that over the course of the next hour. So before we hand across to Marcus, we're going to run our first poll and get to know a little bit about you. So let's get that poll up on screen. So there's that very first question. Speaking of the breadth of range, we'd like to know what your particular background is. Are you working in pharma or biopharma, manufacturing? Do you specifically work in analytics, in industry or academia, or are you a university teacher? Now we're going to give you a little while to answer that, and I can see the answers coming in already. Good range so far. So yes, just answer that. Any questions you have, I can see already there's a couple of questions that have come in. Do get them in at that questions box at the bottom. If you have any concerns about the webinar itself, so if you're not seeing things, if you don't hear things and so on, you can also ask those questions there. And one of my team, in particular Frances Addison, is going to do her best to make sure that we sort this out for you and make it the best possible experience. If you have to leave early, if your connection breaks up, if you have any problem, don't worry, we'll send you a link to the recording of this webinar in a couple of days' time, along with your certificates. You should see that soon. I think that's probably about enough of you voted now. Let's have a look and see the actual results from this. So we have a very even spread throughout everything. Analytics and academia very slightly taking the edge over the others there, but by and large, we've got a good range of people with a good range of backgrounds, and we will see how these statistical approaches and the software and the work that Marcus has done will be able to be applied to all of these. So keep your questions coming in. That's quite enough from me now. I'm going to hand over to Marcus. So we'll get Marcus on screen, and we should see his presentation or his screen very shortly. That's the presentation. That's a good start. Let's see if we can get your camera working. Marcus, wonderful. We can see you, we can hear you, we can see your screen. Thank you very much for joining us.
Hi, Ben. Oh, great, thanks. So I'm just going to start to share the screen. Please let me know if for any reason you can't hear me or you can't see the screen.
Looks good to me, and I think I can certainly hear everything. I can see everything. So I will just step back and leave you to it. We'll speak to you again later on. Thanks ever so much.
Okay, great. Okay. So yeah, thanks to Ben. Thanks for the introduction, and thanks indeed to Chemistry World and Jump for the opportunity to speak a little bit today on good practices in data analytics. So we'll get to it. So what we want to learn today, then, we want to go through a little bit up front about who PMTC are, and I guess address the question, what's all this fuss about data analytics, and why should we be thinking about good practices in data analytics? I'll again go through some definitions and considerations on how data analytics can be defined. I'll talk a little bit about data readiness, and specifically then a little bit about structure, data structure. We'll go into an example of that. Come back then from the example and Jump, and we'll talk a little bit about data visualization and go through a case study of that in Jump as well. And we'll round out the session then with highlighting some good practices and considerations around model development. Okay, so PMTC, as Ben said, we are the Pharmaceutical Manufacturing Technology Centre. So we're based out of the University of Limerick in Ireland, and we're based in the Bernal Institute at the University of Limerick. You can see here from this slide that we collaborate far and wide with a number of research institutions, and we have a good range of industry members in our membership, from multinationals to small and medium enterprise. The majority of what we do is industry-led and shaped by industry. So we're very focused on applied research. And as it says here in this mission statement, we're really targeting advanced technology solutions in the biopharma manufacturing sector. So data analytics at PMTC is one of three core themes, research themes for the center. We look at pharmaceutical plant cleaning. We also look at process control and optimization. And then integrated within those two themes and standing alone as its own theme also is the theme of data analytics within the center. So data analytics within PMTC is well aligned to support initiatives around pharmaceutical quality by design, PAT, continuous manufacturing, and the movement towards Pharma 4.0. And we also support our member partners on other advanced analytics initiatives as well. So what's all the fuss? I guess we all know that data is a hot topic, and quick online search for data will quickly reveal a number of interesting metaphors. As data as a resource, it's been likened to oil, soil, gold, sunshine, and even the raw material of business. So there's a lot of hype around data, but there's also tremendous amount of value in the resource of data, and we're set to create a lot more of it. We're set to create 175 trillion gigabytes more of it by 2025, according to some estimates. And this is, if you get a sense of this, I guess it's equivalent to taking Blu-ray discs and stacking them all the way to the moon, and doing that 23 times. That's what 175 trillion gigabytes of data looks like. So it's a very valuable resource, and we're about to create a lot more of it. Recent reports also suggest that a lot of the manufacturing sectors and other sectors are significantly lagging in their use of data or their digital maturity. And so we need to start to be thinking about, how we're going to get the most value out of our data. And certainly when we think about data, we want to be thinking about good practices in data analytics. So in order to kind of address the current challenges around data use and data analytics within the pharmaceutical and biopharma manufacturing sectors, so specifically PMTC have generated a guide to data analytics. And our goal here is really to, as I said, address those challenges around terminology and business and technical challenges, but also it's ultimately just to support and promote the implementation of data and data analytics methodologies in the manufacturing, biopharma and pharma manufacturing sector. So as Ben mentioned at the outset, we've shaped this guide with significant contribution from industry, really, across interviews and case studies and focus groups. There's a lot of variation around terms that can be used around data and data analytics, and so we've defined 51 terms, taking our references there from literature sources and from ISO, International Standard Organization, and pharma regulatory documentation. Represented in the guide is the views of 14 multinationals and SME groups, and our guide largely contains very detailed case studies and interviews. So there's 7 detailed case studies and interviews within the guide. And I'll just touch on those in this slide here next as well. So the case studies and interviews are really multiple data points, if you like, to allow us to extract the good practices and good recommendations in data analytics that I'm going to talk about very shortly. And the guide itself is an 80-page document, but about 45% of the document contains those detailed case studies and interview sessions. As you can see, the case studies are brought forward by a group of number of multinationals and a number of smaller organizations. So we've a good diverse representation of viewpoints there. Also just point briefly as well to one of the interviews in the case study and case studies provided was provided by Jump, where we provide some really good detail around statistical analysis and design of experiments. So let's get into it then. Some of the content in the guide and some considerations around good practices in data analytics. Before we talk about that, let's talk about defining data analytics and what we mean by that. So the question is, isn't data analytics just analyzing data? And so we've had a number of discussions with our industry partners. We've been through the literature, and I guess the short answer is no. It's not just analyzing data. It's really important to consider data analytics as a composite concept, as defined here by the ISO. So we're not just analyzing data. We're collecting it, validating it, processing it, quantifying it, visualizing it, interpreting it, all with a view to moving towards things like predictions and recommendations for next steps. So data analytics and data science are terms that are often used interchangeably. So for clarity here, I've included the definition of data science according to the ISO. And so here we're building on all the work of analytics, and we're incorporating this concept of actionable knowledge and hypothesis testing when we consider the term data science. So that's a little bit our attempt to reconcile the two terms that are often used interchangeably. I'll draw your attention here to the Venn diagram on the right-hand side. And really the way to consider this Venn diagram, I guess, is to read it from the outside in. So if we think of data analytics, we do best to consider it touching on these kind of four main areas of, domain knowledge or expertise. You have computer science and IT aspects to consider. Obviously, we have the mathematical and statistical processing aspects to consider. And more now, we see that there's, entrepreneurship and entrepreneurship qualities to data analytics and data exploration, and all that goes with that. As we move closer towards the center of the Venn diagram, we're hitting on a number of roles that we consider people who will use data analytics and provide very valuable results to support the center of the Venn diagram, which is that decision-making process. So that's a little bit on how we think about defining data analytics. So I'm going to talk for a little bit now as well on why data readiness is important, and a little bit on what we should be thinking about in terms of good practices in terms of data structure. So if we look at this slide, we can kind of read it from left to right. On the left-hand side, we have the situation where we are now today, where a lot of our data sources and our data are disparate, disconnected, and not harmonized. And on the right-hand side, we have Industry 4.0. This is kind of where we want to go, where we have good data visibility, transparency, really good predictive capabilities, and even adaptive capabilities where we can recommend in real time, perhaps even the next steps to take. Okay, so I guess all the work that needs to be done, it lies in the middle here. It lies around, getting your data ready. So cleaning, contextualizing it, formatting it, and getting it ready for machine readability and usability. So that's really the work of today, and this is where we need to start to consider really in detail some of our good practices around data analytics. So when we think more about data structure and good practices and data readiness, we need to think a little bit about the fact that not all data is created equal. Some data sets that are streaming in might be missing values. They might have different units that we need to address. It might just be collected from sources that we're not interested in, or we don't consider the quality of that data good enough. So the acronym GABO, garbage in, garbage out, is kind of highlighted here to make us think about that. Is the data that we're using to build our models and do important stuff and make decisions, is it good enough? Data cleaning is kind of connected to that concept, this idea of, as I said, formatting and arranging data so that it can be analyzed. Oftentimes we're spending more data or more time cleaning data these days than that actually goes into the analysis part. So that's a very important consideration for when we think about good practice in data. We want to be thinking about the quality of data and if it needs cleaning. You can get bogged down in just arranging and sorting the data. It's important to appraise and be aware of the condition of your data, but don't wait to get started, because you can't determine where the value is until you get started and do some proof of concepts here. So by all means, let's think about how we arrange our data, but don't wait to kind of get started and take it to the analysis stage to really start to make decisions and get value from our data. The graphic on the right hand side here is a little bit an illustration of what we might consider to be best practices and how we begin to structure and lay out our data. And so shown here what is just imagine this box here as a table, and the columns of the table are the variables that we're interested in, and the rows are the objects. So variables like time, pressure, temperature, and the objects then might be things like a label or a timestamp and these kind of things. So I'm just going to go through briefly an example in JMP to show that it's really good practice to begin to consider how we lay out our data and that we lay it out in this way. So I'm going to start to show my JMP program here. Please let me know if you can't see my screen, but I believe it should be available there. Okay. So this is a data set that we generated in our lab. Okay, so we have time, and then we have in each column we have the force values generated at each time point for a given tablet that we make on our tablet press. So let's just take a quick look at this data in JMP, and we can begin to use the graph builder. Let's just take a quick look at what we're actually looking at. We look at the first value here. So we can see that we're looking at compression force profiles, and we can begin to build up that to see all of the data sets that we're looking at. So JMP is really useful like this. It allows us to see, okay, so we've got a number of different profiles, and it looks like some of the profiles have different features. And so if we want to look at that in a little bit more detail, let's just look at the individual force readings for each tablet. And so as we go through this, then we might consider, okay, that's nice, but I'd like to, just color the, add a bit of color there on each of the tablets. But in fact, you see, and this is really a function of the way we lay out the data. We see here that, all the data sets are colored the same. They're not colored individually for each tablet. And we're running into a little bit of difficulty here with the marker type. We have some unassigned values, and this is really a function of the tablet data, the force data being different lengths in that table. Okay, so if we come back to our table and we consider, so that graph there is really not what we want. We want the tablets to be colored individually. And if we come back to our table and consider how we've actually laid out the data here, so I've taken my time variable, and then I've taken my force variable for each tablet, and I've simply repeated that force variable. It's the same variable, it's force, but I've repeated it across a number of columns. A much better way to arrange this data is to consider that I have one variable, i.e. force, and then I create a label for each tablet for the corresponding force. So what that really amounts to is actually rearranging and stacking this data, and JMP allows you to do this really easily, really readily. So we use the stack function. We go to tables and stack, and we select all of the tablet data that we're interested in, and we'll just call this stacked data for now. And our data, as we mentioned, is actually force, right? So we're labeling the variable here, yeah, and the units there are kilonewtons. And we're also going to, as I said, create a label column or label variable that will tie each tablet to a given force in the now new stack table. So there's a number of options here, but I'm just going to accept the default options for now. So we can see here that we've rearranged the data, and we have the time column or variable, we now have a new variable which is label for each of the forces, and we have the variable that we're interested in, which is force, right? So we're just going to show what this looks like again when we actually try to graph it using Graph Builder, and you'll notice that this label data type here is now categorical. So this little blue triangle indicates that it's continuous variable, and this red stacked kind of column icon indicates that the variable is categorical variable or nominal variable. So now when we color by label, we can see that this is kind of more what we want, right? We want clear indication of color per the different tablets that we've manufactured. So that gets us thinking a little bit just about the best way to lay out data. We really want to be thinking not in terms of spreadsheet where we might initially just pull in the data in something like Excel and try and plot it as quick as we can. Really try and get to some understanding of what we mean by variables in columns and objects or labels in rows. And so that's just a summary highlight there of some good practices when we consider data structure. And this is very important data structure when we consider more complex data sets which are multivariate in nature, when we want to do more complex machine learning modeling on that data set. It's very important to consider that we lay out the data and structure the data properly to begin with. So I'm going to return now to the slide deck, talk a little bit about data visualization and how that can be used effectively. So this slide here, I call the good and the just bad and ugly. So these are two data sets. These are the same data set, graphed in two very different ways, as you can see. So on the left-hand side here, it's a good representation of the data, or it's good graphing technique. So we've got clear labels on the axis. We've got good use of the legend. We're clearly highlighting the data set that we're interested in, and so the graph on the left here tells a really good story. The graph on the right, we run into all kinds of difficulties because we're using three-dimensional graphing when it's not needed. Our data labels are very small. We're overusing grid lines here. We're repeating the use of the legend when we don't need it. It's on this axis, but we don't want a 3D graph to begin with anyway. So there's all kinds of problems on the right hand side. The guy on the left hand side is kind of where you want to be at. Another important consideration, good practice if you like, in data visualization is really just to ask the question, what does the data look like? So the data set shown here is four data sets generated by Anscombe in the 1970s, but it serves to illustrate the point that if we don't look at our data and we just rely on statistical descriptors, we can run into all kinds of problems, right? So you'll notice that each of these data sets, they each look very, very different. But when you take things like the mean, the variance, the slope, and the correlation between the two data sets, you can actually get the same number across each of these four data sets. So it's quite a powerful example for reasons why we need to continually look at our data set. And we'd advise from a good practice point of view that, when you're exploring data and working with data and transforming it and doing calculations, that you really visualize it and look at it at each step along the way. The other thing to note here is that, real world data sets aren't this conspicuous in terms of their differences. This is leading me to another really important point in good practices in data analytics that we found from all of our work in the guide. It's really important to connect any analysis and work that you do in analytics with subject matter expertise or domain knowledge. So you kind of want to have the questions around, okay, I've done this work, and I have this outlier. And you want to be able to go to your process expert or your domain knowledge expert and say, you know, is this really an outlier? You know, does it make sense to show the data in this way? So it's really important that reliance on knowledge domain, that it be connected with the data analysis. And that was really a strong point that came out from the work of our guide and good practices in data analytics. I'm going to touch just briefly on a case study, some work that we did in our labs at the PMTC, where we were screening for the optimization of an online measurement position for in-process parameters. So I'm going to go through this example in JUMP, but essentially what we have here is illustrated on the right-hand side is the process. We have process parameter 1, process parameter 2 input as inputs, and we have 9 measurement positions in the process. And at each of these 9 measurement positions, we distilled 4 response factors from each of these 9 measurement positions. And we wanted to know, could we begin to screen and build a model for any of these response factors in any of these positions that would predict process parameter 1 or process parameter 2. So there's 12 runs involved. We have 3 levels for process parameter 1 and 2 levels for process parameter 2, and we repeated each measurement. So this is an early screening study, but gives you a sense of, I guess, what we did. And what we're going to show here and kind of explore in JUMP is, you know, how readily we can visualize this relatively complex data set, select and subset the information that we're interested in, and then begin to build a model on that. So I'm just going to show my JUMP screen again, get rid of some of the old screens or the old menus. Okay. All right. Okay. So you can see here that we have all of the data from a case study runs that I just described there. So we have our test label, we have our process parameter 1 at 3 levels, as I mentioned, process parameter 2 at 2 levels. We have 9 positions in the process, and then for each position we have 4 response factors for each of those measurements. So we've adhered to the good practices in terms of structuring and laying out our data. So now we can kind of get into it and explore it a little bit more. So yeah, let's start building it up. You know, Graph Builder is very useful as a function in JUMP to really quickly just put stuff down and start to take a look at it. So we're obviously going to look at process parameter, our inputs, process parameter 1 and process parameter 2 on the x-axis, and our outputs or responses that we're interested in on the y-axis. And as you can see, you can go through it and start to build up quite a lot of data, you know, divide it out. And we can actually even divide this further because, as I said, remember, we have 9 measurement positions that we're screening in the process. And so you can see here, and in this instance, let's just highlight process parameter 1 because that's what we're going to look at first. So you can see that it's relatively quick and easy to, you know, build up the ability to just visualize all of the data using JUMP and using Graph Builder. So lots of dots on the screen, really useful to consult subject matter expertise in this area. One of the things that we noticed initially from the runs was that this Y2 response value here across process parameter 1 and 2 is clearly almost at a level of 0, and this is really an undesirable state, and we rely on process expertise, knowledge domain expertise, really to kind of inform us of these things. So we just want to get rid of this data actually from measurement position 1, 2, and 3. So you can begin to select that data I'm just going to select it up here because it makes it a little bit easier to select it. Okay, so we've selected measurement position 1, 2, and 3, and what we can do with Graph Builder is we can just get rid of that data, right? We can hide and exclude it. So hide it removes it from the graphs and excludes it from any future analysis or measurements that are future data analytics that we might do. So now we want to look at the other relationships and see what's happening here. So we can see that in certain instances, in certain measurement positions for process parameter 1, we get some, you know, kind of trends that are maybe desirable. You know, this kind of, there's different trends there with the different levels for process parameter 1. Draw your attention there to position M8 and the Y2 response. You can see that we have quite a tight response across Y2 for the M8 position. So we've now selected that data, and really good feature of JMP here. You know, we can see that if we go back to the original table, that data set has been, that we've selected on the graph has actually been highlighted on the table. So that's really useful. And you'll see here as well that positions M1, 2, and 3 have all been hid and excluded here. And so that's labeled on the table there. But the fact that these are easily selected on the graph and then correspondingly highlighted on the table makes it very easy for us to do things like just subsetting the data. So we subset the data. And in this instance, we're just going to accept the default. We're going to take all the columns from the M8 position, and here now we already have a subset of the data set that we're really interested in, and we can begin to use the fit model function in JMP to begin building up model. Remember, Y2 was the response, and we're interested in modeling Y2 as a function of our process parameter 1, different levels there. Okay, so we can see that we add in those X and Y responses. I've noticed as well that I've actually left my baseline runs in here as well, so I can highlight that and on the graphs and the output from the model, and I can actually just exclude and hide that data set as well. And then we can just come to, let's just do the fit model again. Really useful button here, depending on the level of complexity that we're dealing with, but you can just recall that previous you know, population of those variables there. Okay, and so let's run the model again now with our baseline points extracted. So we can see that, you know, our residuals here certainly seem to have a bit of curvature, so there's likely a bit of curvature. So we can go back and begin to explore that again in the model. You know, we do recall. Okay, let's look at, you know, a little bit of quadratic or crossing term here, and we just run the model again. Okay, so you can very quickly see that, you know, we've improved our distribution of residuals there, and we've got an early screening model here with a pretty good R squared value. There's other features here as well, and I think I can just highlight briefly here where we can do some things like you can just do quickly interpolation using the prediction profiler, and that gives us predictions for Y2 with a process parameter 1, and our confidence intervals are highlighted there. So at this point, I will hand over to Volker for some maybe other examples of this, and we might do that via Ben or other folks.
Certainly. Well, yes, thank you ever so much, Marcus. Fascinating stuff so far. We've got a few decent questions in so far, along with a couple of my own. But what we'll do now is we'll hand over to Volker, who you can now see on screen, and we can see Volker's screen. Let's just check if we can hear you. Volker, welcome. Thank you for joining us.
Thank you, Ben, and Marcus, for your inspiring presentation. And before I hand back to Marcus for a wrap up, I want to add 2 other good practices in data analytics by referring to our customer success story from Novomer. This data table shows their multivariate problem at a later stage. Using exploratory analysis and data mining in JUMP, they already reduced 35 explanatory variables to this short list of 10 predictors. And the goal is to find the process settings giving us the highest yield. The other goals like minimizing an unwanted byproduct at the same time are not discussed here. And by the way, the final solution was sold to a spin-off for $100 million US. My first good practice is about visualizing and sharing such a solution as suggested by a statistical model. JUMP's prediction profiler is always recommended to confirm or enhance your subject matter knowledge and to get a deeper understanding about your product or process. Here we can see three main effects: temperature, monomer, and water, a quadratic effect causing some curvature here, and also a two-factor interaction. And as Markus said, don't just look at summary statistics, also look at the data. I am saying not just look at your estimated parameters and p-values and equations, also look at the prediction profiler and use its interactive visualization to see what's going on. And the profiler can also help you to find the best solution, which is the highest yield we can expect at these settings here. And when you get to these results, for the first time, JUMP also shows some diagnostics like an actual by predicted plot, which looks good in this case, and also residual information you can use and you should use to validate your modeling assumptions. This insight was not possible by analyzing big data, but by collecting exactly the data Novomer needed to solve their problem. They measured yield 21 times by simultaneously changing all their 10 inputs. Following this statistically designed experiment, a definitive screening design in this case, which comes as well with some helpful diagnostics you can check before actually running the experiment. On each run, all the inputs are set to a low, a mid, or high level to measure yield. And after adding the yield values to the data table, experimental data is analysis ready. So you don't need to worry about missing values or multicollinearity. In contrast, observed data can be very messy, as Markus pointed out. And as my second good practice, I really recommend to add design of experiments to your toolbox to learn more and faster. For a discussion of the importance of DOE, especially in pharma manufacturing, please refer to the final chapter in the PMTC Guide to Data Analytics, as mentioned by Markus. You see the title page on the left on my screen. And to learn more about DOE concepts, I suggest this 5-session online workshop on smarter experimentation for chemists, but also for scientists and engineers, which is presented by my colleague, Phil Kay. Here you see what you can learn during 5 sessions, and you can also use the JUMP free trial for the hands-on. And after completing this course, you will also get a certificate. To get to this page, just grab the URL from the chat or simply Google for Chemistry World Smarter Experimentation. For any other question about our resources or for a personal JUMP demo, please contact me at volker.kraft@jmp.com or on LinkedIn. And with that, back to Markus.
Thank you very much, Volker. Let's just get Markus back on screen. I can see we had a few good questions come in during that bit of the presentation as well. We are edging towards 1 o'clock. We will allow the presentation and the Q&A to overrun a little bit just to try and make sure we can get several of your questions in. So don't worry about it, and please do stick with us after the 1 o'clock mark. But get your questions in anyway, because we do keep a record of them, and they're very useful. And we may be able to get our presenters today to drop you a line if they have the useful answer for you. So I will hand back to Markus, and we'll come to your Q&A not long after that. Thank you ever so much.
Thanks, Ben. Can you see and hear me?
Yes, we can. All good.
Great, thanks. Okay, so just to round out then the last 2 or 3 minutes here, just want to hit on some good practices to be considered around model development. And we're seeing there in the examples that I went through and also what Volker touched on. Oftentimes we need to think about residuals, you know, and they're really a key cornerstone when it comes to evaluating and developing your model. So just shown here on the right hand side some illustration of residuals, and obviously the upper 2 plots here on the right hand side of the slide are less desirable. You know, we don't like to see patterns in our residuals. We don't like to see you know, differences in the data around 0 for the residuals. So more desirable is the graph on the bottom right shown here, where we're randomly distributed. Our residuals are randomly distributed and approaching normality with larger data sets. So I've just listed on the left-hand side here some points to consider, you know, when we're building up the models, and we can do this, you know, at the practitioner level, and we can do this at the kind of higher level management level as well. You know, we need to have those dialogues. We can consider things like around, you know, data set selection and size. Are we covering the ranges we need? Do we have enough data to be statistically confident? You know, are we exploring our data for any correlated inputs? It's going to have an effect on our modeling output. Have we made assumptions of linearity, and are those assumptions valid? You know, are we touching base with our subject matter experts, our domain knowledge experts here to kind of understand that? Other points around, you know, training the data set, validating it and improving it, you know, by dividing up the data set or collecting separate data sets, and then ultimately testing it with a further independent data set before moving forward with our model. You know, we want to be selecting variables, curating our variable selection And we also want to be considering, you know, how accurate do we need to be? You know, what's our limit of detection? How precise do we need to be? Do we understand outliers fully? So all of these points and more are covered in our Guide to Data Analytics for Pharmaceutical Manufacturing from the PMTC. And so we are making the guide available to download online. That's part of our new website coming in March 2021. In the meantime, if you want to, you know, get some more information on the guide or more information about data analytics and the other work we do at PMTC, please email us at pmtc@youll.ie. I'd just like to acknowledge all of the case study and guide contributors throughout the work of the guide, and also the PMTC team at Limerick. Also acknowledge our funding as coming from Enterprise Ireland and our hosts at the University of Limerick. And thanks once again to JUMP and Chemistry World for the opportunity to speak today.
Well, thank you very much. Thank you for joining us. It's a fascinating 50 minutes so far. We do still have time for Q&A. It's really interesting to see how these sorts of quite specific observations that you've made can be applied to different industries. Before we actually get into the Q&A, we're going to run our second poll for today. So let's get that poll up on screen now. So this will help us to shape future webinars as well as help us to feedback to JUMP. So you find it easiest to learn about data analytics by watching demonstration seminars and videos like this, reading about case study examples, practicing the techniques with example data sets, practicing the techniques with your own data sets, or something else. So do please let us know, because that will then help us know that maybe we need to do more hands-on practicing sessions like the one that Volker was just talking about that's coming up later this month. Now, in particular, that one is geared towards, in terms of time zone, is probably best for people in the US. If you think that's going to be too difficult for you to get to because of the time, then do drop us a line, cwwebinars@rsc.org, and we will see if we can arrange another series later in the year that will be in a more comfortable time zone for you and really allow you to make the most of the interactivity with JUMP staff who will be live in there to help answer your questions and to deal with issues that you may have. Let's just give it a little bit longer for more people to vote in that, then we'll have a quick look at the results before coming through to the Q&A session. One other thing that Volker didn't mention that might be useful, we are running a webinar roughly once a month with JUMP looking at different case studies, different examples, different ways to use these statistical techniques and the software that comes with it. So have a look at chemistryworld.com/webinars and you will be able to see for example, the next one coming up roughly this time next month, 16th of March at 11 AM in UK time, is on gaining an edge through smarter experimentation. And in that case, we are working with Timothy Gardner, who's the founder and CEO of Riffian, and we're looking at how they are able to use these sorts of techniques. So it might be that maybe it's just closer to your industry, or you just want to see another example of how this can work really well before you choose to take the step, in which case pretty much every month there'll be a JUMP webinar that you can join us for, and please do. They're always interesting, they're always enlightening, so it's really good to have as many of you with us as we can. So that's probably enough time for the poll. Let's get that closed down and have a quick look at the results, although it's more for us than for you. Well, clearly 62% of you enjoyed what you saw today and found that this was a useful way of learning about it, but that practicing techniques with example data sets is a key one. There is sample data available as part of that week-long course. So if that's how you learn best, do please sign up for the course and we hope to see you there. Right, let's get on to some Q&A in that case. Let's get Volker back on screen as well so that he can chip in as and when. Marcus, one thing you mentioned earlier as being really important was about data cleaning. What do you think, or what in your experience have been the main types of data cleaning issues or the most time-consuming data issues that people are likely to encounter?
Yeah, it's a good question. So I guess it depends a lot on the data set, but typically you're dealing with missing values or incorrect units quite often. You have to rearrange the data in the best way. you know, you might run into time-consuming activities if you need to, you know, create summary statistics where you're condensing a large amount of data to give a single value so you can make a comparison between a number of variables. Those types of activities take a bit longer, and you know, if you're doing this then in industry, you know, getting alignment on those kinds of activities across a number of functions can also be quite time consuming.
It's certainly the case with a lot of this. I think that people have their own way of doing things within their group, within their team, and that may not match up with somewhere else in the same company, in the same building. And as a result, there's a lot of data out there that just isn't useful enough and could be so valuable to us if we just took these few important steps. One thing I was wondering, you, as part of the process for building these guidelines, you clearly spoke to a lot of different people. Are there different types of industry, different types of research that have specific problems that they should address first? Or do we find that basically data is data, everybody has essentially the same issues that can be resolved by following these best practices?
Yeah, I think there's, the answer is yes and no. I would say, you know, yes, there are general concepts in data analytics where you have good practices that hold. I think we find, particularly on the pharmaceutical biopharma side, you know, regulation is the big elephant in the room, and you know, there are nuances and things to be teased out on that front, you know, that might be specific to the biopharma manufacturing sector. It's very heavily regulated. So yes, there is some general stuff, but then, you know, when you start to break it down per industry, certainly the biopharma industry, the asks can be more specific when it comes to good practices.
Of course, in that case, it's mostly about reporting that data, isn't it? And so having data that you know is clean, you know is reliable, you know you can machine read in future. If regulation changes, you need to report new and different things based on historical data. By having your data tidy, accessible, machine readable, you've already solved a lot of those problems.
Certainly, yes. Yeah.
Volker, let's bring you in here. We've had a question from Navid. Just as an overview, could you briefly explain what the scope is for data analytics in their case for an undergraduate student? So I guess, is this a useful thing to start learning now, very early on in a career? And how can it be applied throughout the rest of a career?
Oh, that's a good question, and the scope is maybe the main scope is really using the data to understand and to increase your understanding about the world or your process, your product, typically the world in an undergrad course, and then also sharing that information and using it for good discussions, share it with other people, typically non-statisticians or other people who are not data savvy, but who are interested in a certain scenario or problem, and to share what the data is telling you with others, with subject matter experts, to enable them to make better decisions. And yeah, that's maybe the high level story about data analytics. And then, yeah, it gets down into certain application areas and fields. And if it's your area, if it's your field, and if these are your problems, I think then it's really, really excited to see how data analytics can really help you to move ahead and make better decisions.
Because we're not just talking about business decisions, are we? For an undergraduate, being able to do proper data analytics will help with reporting and help with writing reports for their supervisor. For an SME, if you're better able to communicate what your data can tell you, you're more likely to be able to attract investment. For somewhere in industry, again, if you can present this better, you can take this to your senior management and say, look, we believe this change needs to be made. And having it visualized like this just makes it a lot easier to get people on board and make the changes that you need. We've had a question here from Owen. Owen, it's quite a wide question, but I'm just going to sort of pick the middle bit out of that. And I think we'll start with Marcus, but Volker, I'm sure you'll have something to add as well. What recommendations would you have for handling observational data that has missing data points for one or more variable? Do you just ditch that entire variable, or are there other tools, other ways to infer what would be there?
Yes, so perhaps Volker can talk on the tools front, but I suppose from a point of view of awareness of your data, I would say that it's important to kind of know where you removed the data from, and have that information. No matter what you decide to do with it, having a kind of a track record of where it's been and where it's gone, and that from a kind of a regulation perspective, if you feel like the farmer perspective, is always useful to know, and in some instances really critical.
Almost like the chain of custody in forensic evidence, you have the same thing for data as it moves through different departments of a company.
Yeah, I think so. Recommended. Yeah, I think so. Yeah.
So Volker, any advice on handling missing data points?
What I can say is it's very similar to outliers. It's not an automatic process. So like with outliers, what to do with them? Correct them, exclude them, or ignore them, or just keep them. Same with missing values. What to do? Does it help to impute maybe averages, or to fill the gaps somehow? What could be done in JUMP as well? Maybe the missingness is also carrying some information. Then it would be good to use this also for building predictive models, because this information can really help you to get a better model. So it depends, but all of this needs thinking of the user, what to do and what is the application context and what's the goal of the study.
Excellent. Well, we've already overrun. Most of the audience have very kindly stuck with us, but I think we probably should draw it to a close there. So thank you ever so much, Marcus and Volker, for joining us today. Really interesting presentation. I hope that all of our audience will take advantage of the guidelines as and when they're released on the website we have. I think we'll be sharing the information as to how to find those. So clearly there's a lot to learn from those. You've done the legwork of finding out from industry and academia what it is that we need to do to get better at data handling. So thank you ever so much once again. So that is it for this Chemistry World webinar. As I said, this time next month, there'll be another webinar that we will also be running with JUMP, and we can look at another example of how these statistical approaches and the software that they create in order to enable you to use those will really help you, regardless of whether you're in industry, in academia, in education. There are things that you can get from handling your data properly. So I look forward to seeing you in a month's time. chemistryworld.com/webinars to find out the information about that. Also, that's where you'll find the information about the 5-day JUMP course that you can join us for in March. It's being run again in April as well, if you find that you're not able to in March. The certificate for everybody who's attended live will be with you in a few days' time. Likewise, a link to the recording in case you wanted to review any of this presentation or the demonstrations again. That will be with you in your email in a few days. I'm Ben Valsler, Chemistry World's digital editor. I'd like to say thank you once again for joining us for this Chemistry World webinar.